Today we're going to be talking about phosphorus, the nutrient that stores, moves, and controls energy throughout all of our plants. We're going to be talking about what phosphorus does in the plant, how it's taken up by the plant, and how it interacts with other nutrients and moves in the soil. The first function of phosphorus is its involvement in the universal energy currency of ATP. We're going to get a little complex, but it's super interesting, so bear with me. So ATP, adenosine triphosphate. That means we've got 3 phosphate groups. So check this out. We got 1 phosphorus, 4 oxygens. 1 phosphorus and 4 oxygens. Notice how they share this oxygen and that all of these phosphate groups are negative. Basically, that's like saying we have 3 negative sides of a magnet, but they're being held together. When they're being held together, we have this bond right here. That is what holds all of the energy of ATP. There's a tension, like a spring being compressed. Like imagine holding 2 magnets that have the same charge together. They're constantly pushing back. So we have a lot of this potential energy. And what happens is whenever a plant needs something to happen, it'll grab the ATP, and this bond with a bunch of energy will pop off, and it releases a bunch of energy. And what that does is it helps catalyze enzymes and is pretty much responsible for all of the energy needed to power all of our plant's functions. For the second function of phosphorus, we're going to talk about sugar metabolism. Basically how the plant turns sugars it created through photosynthesis into that ATP that can then be moved around the plant to provide the energy for all plant functions. So we're going to go over a couple key steps on how we get ATP from sunlight. So the sun that hits our plants breathe in CO2 plus water they get from the roots, and then we go through the photosynthesis process and we get sugars. Now, these sugars, they accumulate in the leaves, but in order for our plant to use the energy that's stored in these sugars, this glucose molecule, the sugar, needs to go through another process. So we're going to bring it back here. So we got glucose, and it's going to grab on to what's called a phosphate ester, so another phosphorus. It's going to go through this process called pyruvate, and then from there, it gets turned into ATP. So this brings us to a super interesting fun fact. When you have not enough phosphorus in your plants and the older leaves get all purple, that purpling comes because those leaves have created so many sugars in them through photosynthesis, and those sugars haven't turned into ATP. So there's a buildup of these sugars, but those sugars, they're kind of crazy. They don't like to be just stored in the leaves. They need to be turned into this ATP. So to prevent this buildup of these sugars from causing any damage, the plant will release anthocyanins, which are antioxidants. They prevent the oxidative damage from happening to the plant, and those anthocyanins are purple. When we have a phosphorus deficiency, we have a decrease in the amount of sugars being turned into ATP, and that buildup of those sugars can cause damage to our leaves. So to prevent that damage, the plant releases anthocyanins, and that's what turns our leaves purple. So if we don't have enough phosphorus, our plant can't efficiently turn all of the sugars that it's making through photosynthesis into actual usable energy for the plant to grow. And this ATP is responsible for all plant functions. So all of the growing from the shoot tips to the root tips, to making your leaves, to making the fruits, to all kinds of growth requires this ATP. And if we don't have phosphorus, it doesn't matter how efficient our photosynthesis is. If the sugar can't get turned into the ATP with the phosphorus, it doesn't really matter. We just get an accumulation of sugars and it can actually harm our plant. Phosphorus is what lets our plants turn photosynthesis into actual biomass, which is through the ATP. The third function we're going to talk about is how phosphorus plays a key role in the makeup of DNA and RNA. So phosphorus creates the backbone of all of the instructions that every single plant cell has. Without phosphorus, we don't get DNA and RNA, basically the building blocks of all life. Here we have poorly drawn DNA. And phosphorus is a key component of the black marker. It's called the phosphate backbone. Basically, it's what holds the DNA together. If we didn't have phosphorus, we wouldn't have DNA, and our plants would have no idea what to do, because in this DNA is the instructions for every cell to perform their exact function. Every time a cell divides, like at the tip of the shoot or at the tip of the root, when we're having more and more cells, when we're having cell division, that's where this DNA comes in, because every time we have a new cell, it needs to be told what to do, and the phosphorus makes sure that DNA functions how it should and tells that cell exactly what to do. The next function we're going to talk about is the phospholipid bilayer. In every single cell, it prevents certain things from getting in or leaving when they're not supposed to. And so the way it does this is through having the head of each of these layers. So it's a bilayer. We have one layer, two layers, being polar, meaning having a negative charge. Remember this phosphate group that we're talking about has a negative charge? That means this outside that's negative is going to be hydrophilic, meaning water attracting, and this inside is going to be hydrophobic, meaning water repelling. And that happens because of these charges, because the inside of this phospholipid bilayer is a lipid, meaning a fat, so a fatty acid, and so that makes sure that only certain things can enter the cell, and it prevents certain things from exiting the cell. And so if we didn't have this phospholipid bilayer, then we would have a leaky cell membrane, meaning the nutrients that we really need are going to accidentally get out of the cell, and then that cell will die. So when we don't have enough phosphorus, we get, instead of a phospholipid bilayer, we get a different type of lipid. It's just weaker, and it doesn't have the same properties that this phosphorus does, and it'll accidentally let nutrients out of the cell or let things that shouldn't come in. Also, when your plant undergoes stress like heat stress or drought stress, this bilayer having the phosphorus here makes it way more stable under those stressful conditions and prevents the cell from dying under intense heat or drought stress. The last function we're going to talk about that phosphorus is responsible for is enzyme signaling. So we can't have all plant functions be turned on at the same time. It'd use too much energy and the plant wouldn't function efficiently. So it turns certain functions on or off. So like when to start growing or when to release a stress response. Phosphorus is basically like the switchboard that turns them on or off. So we have these phosphate groups. It attaches to the enzyme to turn it on or off. That's really important when we need our plant to react to certain things. So if there's a stress response, we need specific enzymes to turn on immediately or turn off immediately to open up the stomata. So the sun comes out and the plant starts to breathe. To open up all the stomata, we need to have an enzyme signaling using one of those phosphate groups. And so when you don't have enough phosphorus, your plant's not going to know exactly what to do at exactly the right time, because those phosphate groups won't be there to turn on and off certain plant functions. Now we're going to talk about phosphorus uptake. And when we think of phosphorus uptake, we think about phosphorus differently than we think of other nutrients. So when it's taken up by the plant, it's taken up through this orthophosphate form. So we have the phosphate group here, right? And an orthophosphate is just adding a hydrogen to it. And the hydrogens have one positive, so instead of a 3, we're gonna have a 2, and then sometimes we have 2 hydrogens. So this is the orthophosphate. So that's how it's taken up by the plant. But it takes a bunch of different steps, and it's pretty complex on how we get to this stage, because most of the time it's not in that stage, and that's why it's so hard for our plants to access it. So phosphate is negative, and there are a bunch of positively charged nutrients that are in our soil that can't wait to grab onto a phosphorus and not let it go. In acidic soils, phosphate binds to iron and aluminum because there are more iron and aluminum ions available in the soil under acidic conditions. So this PO4 3-, this 3-, and this 3+, or this 3+, they stick together like a magnet and they produce a super strong bond. When phosphate binds to one of these elements, it's a significantly stronger bond. It's more like a handshake instead of a high five compared to, say, a nutrient that's bound to a clay particle. So normally we have calcium or magnesium, and we have clay. Clay has negative, calcium, magnesium have positive, right? These bind, and it's pretty weak. It's just binding on the outer valence electrons, basically the outer shell of these ions, but phosphate's different. It binds on the inner shell of the electrons, so one step in, meaning it locks itself. And when phosphate is bound to calcium, iron, or aluminum, it's bound super tightly. So for phosphate here, we get iron phosphate. This is an insoluble compound that our plant cannot take up, and that binding is way stronger than this binding between calcium and a clay particle. When phosphate binds to calcium, we get Ca3PO42, so calcium phosphate, also an insoluble compound. Plants cannot take it up. Now, there are tons of these ions throughout our soil. Again, when the pH is low in an acidic condition, we have a ton of these iron and aluminum ions. And when our pH is too high in an alkaline soil, we have a ton of these free calcium ions. And these love to bind with phosphate and prevent our plants from being able to take up any phosphorus. But we have a host of mechanisms that our plants have evolved to overcome getting past these super bound insoluble compounds. So our plants need to grab this phosphate ion through the roots. But this phosphate ion is bound to this calcium ion, creating calcium phosphate, and it cannot be taken up by our roots. It also doesn't move very well in the soil. It's going to stay right here. So our roots need to move to the calcium phosphate ion, and then they need to exude an acid. This acid is going to break these apart, and then this phosphate ion is going to go up the root and into the plant. But these compounds don't move very far, right? They move tiny amounts, meaning our roots need to grow to the phosphorus rather than be able to suck the phosphorus up through water, like how it does for sulfate and nitrates and most of our other nutrients that our plants need. So the plant has a couple mechanisms to make this easier. First, when the plant notices that there's not enough phosphorus around, it'll promote tiny little hairs on our roots to where if there's any phosphorus around, it increases the surface area of our roots so it can find and grab that phosphate way easier. It doesn't do that if there's already a ton of phosphorus in the soil. It won't signal that these root hairs are needed. So there's a tricky balance that needs to happen. If we apply too much phosphorus, then we don't get that signaling that we need to develop root hairs, and then we don't get as much nutrient uptake. But if there's not enough phosphorus, then regardless of the amount of root hairs, we're just still not going to get that phosphorus because it's not present. So it's a really tricky balance. But that's where this next concept comes into play, mycorrhizal symbiosis, which is when we have fungi that attach to our roots and they increase the surface area and what we can get from our roots by like 100 to 1,000 times what we normally could. So all of the phosphorus that the plant roots alone couldn't get because it was down here, now the plant can grab them with this mycorrhizal symbiosis. However, there's another tricky thing is when there's an abundance of phosphorus in the soil, the mycorrhizal symbiosis, those fungi, they're told they're not needed. The plant doesn't exude the chemicals that tell those mycorrhizal fungi to attach to the roots and get the phosphorus. So these mycorrhizal fungi exude the acids that break apart the chemical bonds. So if we have like an iron phosphate, the mycorrhizal fungi will come in and they'll chop off this phosphorus and take it up. And it'll probably take up the iron too. But it makes it go from an insoluble compound. It breaks that really, really strong bond that phosphate has with iron, aluminum, or calcium, and it breaks that apart and allows these nutrients to now be accessed by our plant. And so having an abundance of arbuscular, meaning in the root, like attaching to the root, mycorrhizal fungi is incredibly important if we want to have a hyper-efficient cropping system where our crops can have just a 10 to 100-fold increase in nutrient access. And that only happens if we have the right amount of phosphorus in our soils. In a standard soil test, you generally start to see a decrease in mycorrhizal symbiosis above, say, like a 30 to 40 ppm on like a Mylic 3 test. Once you go higher than that, so if you've been applying phosphorus every single season for the past 30 years, it might be difficult to access, and so you may want to do something like an inoculation where you inoculate the seeds with the mycorrhizae ahead of planting So then when those seeds go into the ground, they already have the mycorrhizae and they can immediately find phosphorus. And that's super important for just the entire season of growth because more mycorrhizae mean you have more access to nutrients. So phosphorus moves through the soil through this process called diffusion, but it doesn't really actually move because it only diffuses or moves in the soil 0.1 to 1 millimeter, meaning your roots need to be up to 1 millimeter away from a phosphate ion for your plant to be able to access it. So if you have a root hair that's in this range of 0.1 to 1 millimeter away from the root hair, that phosphate ion can move to the root. But further than that, it's gonna have a really hard time moving at all. Generally, we think of phosphorus as immobile in the soil, and we need our plant roots to grow to the phosphorus, which is different than most of the other nutrients, like potassium, sulfur, and nitrogen, where our plant is taking up water, and with that water, it's taking up all of those nutrients. That doesn't happen with phosphorus because phosphorus isn't soluble, so it doesn't get dissolved in water. And so our roots have to move to the phosphorus rather than pulling the phosphorus up as our plants respire and bring water up into the roots. So how do we know if our plants are getting enough phosphorus, right? We have step one, which is soil test, right? But like we just talked about, phosphorus is heavily immobile in the soil, so even if you take a soil test and it could be adequate, we also want to be following up with a tissue test or a sap test. And I want to talk about sap tests real quick. So earlier we were talking about why phosphorus deficiencies cause those lower leaves to be purple when those sugars accumulate in those leaves, because phosphorus is in control of turning those sugars into usable energy. And the purpling is the anthocyanins that protect the leaves from sugar accumulation. When that's happened, we've already undergone a deficiency stress. And so we can use a sap test to measure the old leaf and the new leaf. And because phosphorus is mobile in the plant, it can be moved from those old leaves to the new leaves. We can see if we have enough phosphorus being taken up by our roots throughout the season if we're taking these sap tests, because we can measure the old leaf and the new leaf. And we can see kind of how much phosphorus is left in our old leaves. And if those are too low, even before we start seeing purple, as soon as that plant goes through a period of rapid growth where it needs a lot of phosphorus, if our reserves in our older leaves are already low, then we're going to have deficiency stress and a yield drop. And so that's why sap testing throughout the season is really cool, because we can pretty much figure out if we're going to be deficient before we actually see those signs, and we can correct them. Now for phosphorus in particular, I'm going to talk about another test. I want to talk about the Haney test and the PLFA test, specifically regarding phosphorus. Now, the Haney test measures for orthophosphate, if you're getting it from the Regen Ag Labs Haney test. It also measures carbon dioxide respiration. So that's pretty much a proxy for how many microbes are in your soil. And the more microbes that are in your soil, the more available phosphorus you'll have available to your plant to uptake. But if we want to get even deeper into our phosphorus availability, we'd want to combine a soil test with a PLFA test, because a PLFA test will tell us how much fungi we have. And so if we have a low amount of fungi, even if our soil test is adequate, we're probably going to see a decrease of phosphorus in our tissue and plant sap. So we can know through a PLFA test if we have an abundance of fungi present that can make the phosphorus from our soil test available to our plants. And so using all of these tests, we can figure out exactly where to put our fertilizer dollars. Whether we should do an inoculation because we have super low fungi, whether we should be adding food for microbial life through certain biological amendments to increase our CO2C, our respiration rates, or if these are all high, but we just don't have that much phosphorus available, maybe then we apply phosphorus granular fertilizer at the root zone in a banded method. And then if it's in the season and we're taking sap tests in the season and we see that we've got a good amount of phosphorus in our new leaves, but our old leaves over the past month have begun to decline, meaning we're not getting phosphorus as fast as our plant is using that phosphorus, then we may want to do an in-season foliar application, and we'd figure that out by doing the sap test or tissue test. And so that's kind of how we can use a suite of tests to figure out the most efficient way to fix a potential phosphorus deficiency. Phosphorus is incredibly difficult to manage because it has those properties that are different than the other nutrients and are the reason that phosphorus has so many functions in the plant, because it's so unique in how it's formed chemically, like with the 3-. But at the same time, that creates more issues like phosphorus getting bound up and not being mobile in the soil as well. And so we can use all of these tests to figure out exactly what we should do to manage our phosphorus. Again, I'm Jack, the CEO and founder of Cultra, a nutrient analytics platform, where you can take all of these tests, put them into our software, and track whether they're going up, they're going down, and exactly where you should put your fertilizer dollars in order to maximize the efficiency of your farm. Thank you so much for watching. I hope you enjoyed this video.